Welcome back. In this section, we'll discuss the early embryonic events which contributes to the journey of a single cell into a multicellular organism. Shortly after fertilization, the fertilized egg or the zygote, it undergoes a series of rapid division to form a ball or cell called morula. The cell division takes place without any increase in size. Such kind of cell division is known as cleavage. So a single cell undergoes a series of cell division to form a ball of cells known as morula. Now the fluid seeps into the morula and its cell realign to form a fluid-filled hollow ball. This structure is now known as the blastocyst. There are two types of population that can be seen in a blastocyst. You have a small cluster within the cavity called your inner cell mass, whereas you have your outer trophoblast cells that line the fluid-filled cavity. The inner cell mass develops into a proper embryo, whereas the trophoblast cells are associated with the implantation of the embryo and formation of placenta. At the end of first week, the blastocyst gets adhered to the endometrium and later gets implanted to the uterus wall. In the second week, the inner cell mass gets differentiated into a two-layer disc called bilaminar embryonic disc. The bilaminar disc is comprised of a dorsally situated columnar cell layer known as the epiblast layer and a cuboidal cell layer on the ventral aspect known as the hypoblast layer. Now, for the sake of understanding, let us assume take a cross section of your bilaminar disc. Now, as the name suggests, bilaminar, it is made up of two layers. You have one layer which is marked by blue color, and underneath that there's one more layer which is marked in yellow. Your dorsal layer is called an epiblast layer, and on the ventral aspect you have your hypoblast layer. During third week, the bilaminar embryo is converted into a trilaminar embryo by a process called gastrulation. Gastrulation begins with the formation of a structure called primitive streak that develops in the midline of epiblast towards a caudal end. This structure is a narrow groove with slight bulging area on each side. And there is a small depression called primitive node at its cranial end. At this stage, some cells of epiblast migrate and invaginate in the primitive streak and primitive node. Some of the cells displace the hypoblast layer to form the embryonic endoderm. Other cells remain between the endoderm and epiblast to form the embryonic mesoderm. The remaining cell in the epiblast produce the ectoderm, and thus the three germ layers are formed. In this way, all three embryonic germ layers are formed from the epiblast layer. Next, coming to neurulation. The first event of neurulation is the formation of a thickened area of cells called neural plate. The neural plate forms in the cranial end of the embryo and grows in a cranial to caudal direction. The cranial or the head end of the neural plate indicates the region of future brain, and the narrower caudal or the tail end represents the future region of spinal cord. By the end of third week of development, lateral edges of neural plate become elevated and move together to form neural fold. The resultant space created due to folding of neural plate is called a neural groove. The neural fold fuse together and neural plate transformed to neural tube, which is a precursor to central nervous system. The fusion of the neural tube usually begins in the middle of embryo, extending in both cranial and caudal directions. During closure of the neural tube, cells on the crest of neural fold detach, forming a new cell population called neural crest cells. These cells contribute to formation of peripheral nervous system. Once the neural tube has completely fused, the process of neurulation is complete.